we're still looking at microscopic anatomy of the muscle cell, and I left off with the arrangements of the myofilaments. And my myofilaments here are the little dots on the inside here, the myofibrils, we're calling them. But anyway, so there's my myofilaments. There's my heavy filament, that's myosin, and then there's my thin one, that's actin. So it kind of gives you an idea of where we're at and what we're looking at. So what we're looking at now is the sarcoplasmic reticulum, or the SR. The SR is all this blue on the outside of my muscle cell. So the sarcoplasmic reticulum is an elaborate, smooth endoplasmic reticulum system. This runs mostly longitudinally, and it surrounds each of the myofibrils. Now you can see on the end, it comes down to an end, and we've got this area here. This is called the terminal cisternae, or cisterna, depends on how you want to spell it. See, there's the word right there. So it's this, and then it's this right here, okay? Now, all of this functions in the regulation of intracellular calcium. Calcium is kind of the key when it comes to getting your muscle cells to contract. Along with the terminal cisterna, we have what are called the T-tubules, and these T-tubules, they penetrate into the cell's interior at each of the A-band, I-band junctions. A T-tubule is associated with a pair of terminal cisternae, and this forms what's called a triad. And I'm looking for it. There it is right there. There's the triad showing the 3. Now the T-tubules themselves, the yellow here, these are continuous with your sarcolemma, sarcolemma being the membrane of the muscle cell. So these conduct impulses. So we're trying to get nerve impulses into the deepest regions of the muscle. And then these impulses signal for the release of calcium from the terminal cisternae. So the T-tubules and the SR provide tight-linked signals for muscle contractions. The T-tubules, there's proteins in there that act as voltage sensors. And then the SR foot proteins are the receptors that regulate the calcium that's going to be released from the ends of the sarcoplasmic reticulum. Now, in the sliding filament theory, which is why we need the calcium, the thin filament slides past the thick ones, so that the actin and myosin fibers need to overlap a greater degree. So I'm going to go back one slide, maybe. Back one slide. So here's my actin, here's my myosin. And when it comes to the sliding filament, we take this thin filament here and we move it closer to our M line up here. So this one on my right moves to the left, and vice versa over here, this one moves this direction. So anyway, in a relaxed state though, you still have some overlapping between your thin and your thick filaments. If you didn't have that overlapping, you couldn't actually get these heads to attach to the actin fiber, which means you wouldn't get any muscle contraction at all. So it's upon that stimulation, these myosin heads, they bind to the actin, and that's when we get these myosin to start pulling on this actin. And so that's where they get the sliding filament. So each myosin head is going to bind and detach several times during a contraction. It acts like a ratchet, reaching up, grabbing and pulling, and then letting go, and reaching up, grabbing and pulling and letting go. And this generates tension and propels that thin filament to the center of the sarcomere. This event occurs throughout the sarcomeres, and that's how our muscles actually shorten. So in order to contract the skeletal muscle, first, it must be stimulated by a nerve ending. So here's my nerve ending coming down from my nerve. From there, we have to propagate an electrical current. It's also known as an action potential. So if I use the word action potential, I'm just talking about that nerve stimulus, okay? But it's along the sarcolemma, so it's on the surface up here. So I get an action potential up here, and then going down the T-tubules, going out here, down the T tubules there, all along the sarcolemma. Anyway, we have to see a rise in intracellular calcium, and that's the final trigger for contraction. So linking the electrical signal to the contraction is called excitation coupling, or excuse me, excitation contraction coupling. Sorry, I missed a word in there. So skeletal muscles are stimulated by motor neurons of the somatic nervous system. We'll talk about somatic versus autonomic when we get into the nervous systems. Axons of these nerves travel in the big nerve bundles to the muscle cells. And then the axon of motor neurons branch profusely once they enter the muscle. So you got all these branches going out. And we end up with these terminal axon terminals going out. So you can see I have a whole bunch of those, okay? So each axon branch forms what's called a neuromuscular junction with a single muscle fiber. Now, the neuromuscular junction is formed when axon endings, which have that, they have small membranous sacs or vesicles inside here, the green ones. They contain a chemical called a neurotransmitter. This neurotransmitter in skeletal muscle is called acetylcholine, acetylcholine. We use ACH as an abbreviation. So each of those vesicles, they contain molecules of ACH, and there's my molecules of ACH. So across the way on the muscle itself, because this is all nerve up here, this is my muscle. This muscle part down here is called a motor end plate. So a motor end plate is where it's a specific part of the sarcolemma. It contains the ACH receptors. There's my ACH receptors down here. And this whole thing's going to help form this neuromuscular junction. Now you got to remember that at the neuromuscular junction, these two don't actually touch. They're exceedingly close, but they don't touch each other. And this space is called a synaptic cleft. The cleft is between the nerve up here and the muscle down here. Okay, so when a nerve impulse reaches the end of my axon, what happens is voltage-gated ion channels open, and this allows calcium to enter into the cell. Here's my calcium coming into my nerve cell. We're just in the nerve cell now. That calcium, once inside, they don't show it down here, but what it's going to do, it causes these vesicles, these green vesicles here, to fuse with the axonal membrane down here. So you can see how they've come in here, they've fused, and they dump their contents. So that's what the calcium does. It literally causes that fusion. Sorry if you want to go into the details on that. I don't think we even covered that in the nervous system, but we may. Let's look into it later. So anyway, this releases, oh, that fell, chair. This releases ACH into the synaptic cleft, and this is via exocytosis. I think that was a biology word. The ACH then is going to diffuse across that synaptic cleft to the ACH receptors on the sarcolemma, on that motor end plate, and you can see that's what's happening down here. When they bind to the receptors, what happens is we open these gates up, and these gates allow sodium to rush in, potassium then will leave, but this starts an action potential in the muscle. So an action potential we'll talk about here in a little bit, but basically we're trying to change the polarity. Whoops, didn't want that. We're trying to change the polarity on the inside versus the outside of the muscle. Right now, if your muscle's at rest, you have all these positive charges on the outside and negative charges on the inside. We need to flip that. We need positive charges on the inside and the negatives can go on the outside. Now, by the way, the ACH just can't stay there. If it stayed there the whole time, that would keep these things open all the time. And that means you'd have like a cramp. So ACH bound to ACH receptors are quickly destroyed by an enzyme called acetylcholine esterase. There's the word right there, acetylcholine esterase. ACE means it's an enzyme. We'll talk more about that when we get into the nervous system. But this destruction is going to prevent continued muscle fiber contraction in the absence of additional stimuli. So we need to control it with our brain to say, keep sending messages to that muscle to keep it contracted. Okay. I use the term action potential. When it comes to an action potential, I like the way the book puts this. A transient depolarization event that includes polarity reversal of the sarcolemma or nerve cell membrane and a propagation of an action potential along the membrane. Okay, what I said before, we're trying to get the positives from the outside here to the inside. We're trying to make the inside less negative because we've got like chloride and other big anions that all have this negative charge on the inside, and we need to neutralize those and even flood this cell with so much sodium that we end up with a positive charge on the inside if we were to actually to measure it. Okay, so ACH binds the receptors at the motor end plate. This binding actually opens the chemically gated channels. Sodium and potassium diffuse out, and the interior of the sarcolemma becomes less negative, and that's called depolarization. So that's what we're looking for. Initially, this is a local electrical event called an end plate polarization, because remember, this is the motor end plate, so it just happens here on the end plate. Later, though, it ignites an action potential that spreads, it spreads in all directions across the sarcolemma. So that's going to send that charge going everywhere. This is simply showing my action potential moving across the sarcolemma, and what that's doing is changing the polarity inside the cell. And when we change that polarity, we actually allow the cell to be flooded with these calcium ions. So the changing of that, by depolarizing, we make it so we have other gates inside here. They actually open up when we become more positive on the inside of our cell. They're based on how many positive and negative ions we have in there. Works pretty well, doesn't it? Okay, in the excitation contraction coupling, once it's generated the action potential, this is going to be propagated along the sarcolemma. So that's what's happening here. Down the T tubules, triggers the release of calcium from the terminal cisternae. And then here's the cool part. I'm going to go back to that picture I had before. So the calcium, once it's inside here, it's going to bind to this troponin. That's the molecule. Remember, the troponin is actually connected to the tropomyosin. So by binding to this troponin, it rolls the troponin, which causes this tropomyosin to roll off of the active sites. This is the best one I've got. See, calcium comes in here, binds here, these roll, that opens all these sites up, okay? So now my actin active sites are being exposed to my myosin. Now the myosin cross bridges alternately can attach and detach. The thin filament then is going to move towards the center of the sarcomere. Hydrolysis of ATP is going to power this process. And then calcium is removed into the sarcoplasmic reticulum. The tropomyosin blockage is restored, and the muscle fiber then relaxes, and we go back to where we started. So, okay, I'm going to stop this video here because I think that's a lot of good information, and we will pick up the rest in our next video.